Hello, we are Team 3, responsible for UV visible and FTIR. In this experiment, we investigated the acid-base equilibrium between benzoic acid and the benzoate ion, as well as the structural changes caused by variations in pH. Using UV-visible spectroscopy, we observed spectral changes during the conversion of sodium benzoate to benzoic acid under different pH conditions, and confirmed the structural changes using FTIR spectroscopy. Benzoic acid is a representative aromatic carboxylic acid that forms an acid-base equilibrium in aqueous solution. When it loses a proton in water, it exists as the benzoate ion, and this reaction occurs reversibly. The pKa of benzoic acid is approximately 4.2. At low pH, benzoic acid form is dominant, while at high pH, the benzoate ion form becomes predominant. This can be understood as a structural conversion between the carboxylic acid form and the carboxylate form. These structural changes also affect the spectroscopic properties of the molecule. Sodium benzoate is a widely used food preservative, but its antimicrobial activity mainly originates from benzoic acid under acidic conditions. Therefore, understanding the pH-dependent equilibrium between benzoic acid and benzoate ion is important, and we investigated this conversion using UV-visible and FTIR spectroscopy. Based on the structural differences between benzoic acid and benzoate ion, we expected pH-dependent changes in both the UV-visible and FTIR spectra. In particular, shifts in the absorption peak and changes in characteristic functional group vibrations were anticipated as the acid-base equilibrium changed. First, a 1 millimolar sodium benzoate stock solution was prepared. After weighing 0.8 milligrams of sodium benzoate, it was dissolved in 5 milliliters of water. Next, solutions with pH values of 1, 4, 7, and 10 were prepared using 0.1 molar hydrochloric acid and sodium hydroxide solutions. Finally, pH paper was used to confirm that each solution had reached the desired pH. First, the instrument is turned on and allowed to stabilize for about 20 minutes. Next, the Spectra Manager software is launched, and the spectra measurement menu is selected. Experimental conditions can be configured in the Measure Parameter section located at the upper left corner of the screen. In the Information tab, details such as the sample name and username can be entered. Now, we will perform the baseline measurement. First, 2 cuvettes are filled with 2 milliliters of distilled water. They are then placed into the instrument with the transparent sides facing the light path, and the cover is closed. Next, the baseline measurement icon at the upper left corner is selected, and the measure button is pressed to record the baseline. After the baseline measurement is completed, the distilled water in the outer cuvette is removed and replaced with the sample solution. Then, the sample measurement icon is selected. In the comments window, the sample name and operator name are entered, and the OK button is pressed. Once the measurement is finished, the file name and save location can be specified using the Save As option in the File menu, and the data is saved in JWS format. If an Excel-compatible file is needed, the Export function in the File menu can be used to save the data in CSV format. The saved files can then be transferred using a USB drive. Next, for the FTIR measurement, we prepared a sample by converting sodium benzoate into benzoic acid. First, 50 milligrams of sodium benzoate was completely dissolved in 300 microliters of water. Then, 200 microliters of 6 molar hydrochloric acid was added to generate a precipitate. The resulting precipitate was collected by filtration and dried in an oven for 5 minutes to prepare the sample for FTIR measurement. Next, the OMNIC spectroscopy software is launched. From the Experiment Selection menu at the top, Is 50 main sample is chosen. Then, the Set button at the upper left is clicked. In the Collect tab, the number of scans and the resolution are set, and the final format is set to transmittance. Once all settings are confirmed, the OK button is pressed. Before measuring the background spectrum, the crystal surface is carefully cleaned. The Collect Background button at the top of the screen is selected to record the background spectrum. After the background measurement is completed, the sample is placed onto the crystal surface. Then, the Collect Sample button at the upper left is clicked, the sample name is entered, and the measurement is performed. Once the sample measurement is finished, unnecessary data can be removed by clicking the Clear button. In addition, the spectrum can be displayed in transmittance mode by selecting transmittance from the process menu. The data can then be saved using the Save As option in the File menu, and screenshots can be captured and saved as image files using the Screen Capture tool. The exact peak positions can be identified using the Find Peaks button at the top, and the search function allows comparison with library reference data. Finally, the data files can be transferred using a USB drive. First, these results show the changes in UV visible absorbance spectra depending on pH. At pH 1 and pH 4, the maximum absorption peak appeared around 230 nanometers. This can be interpreted as sodium benzoate being protonated under acidic conditions and converted into the benzoic acid form. In contrast, at pH 7 and pH 10, the maximum absorption peak shifted to around 220 nanometers. This suggests that sodium benzoate mainly exists as the benzoate ion under basic conditions. Therefore, these results indicate that the equilibrium between sodium benzoate and benzoic acid changes on pH, accompanied by a red shift in the UV visible spectrum and a decrease in absorbance intensity. Next are the FTIR measurement results. First, we compared the FTIR spectra of benzoic acid and sodium benzoate. In the spectrum of benzoic acid, a broad OH bond peak appeared in the region around 2,500 to 3,300 reciprocal centimeters, along with a CO double bond peak near 16,8-1710 reciprocal centimeters. In contrast, these peaks disappeared in the spectrum of sodium benzoate. Instead, characteristic carboxylate peaks were observed around 1,550 to 1,610 reciprocal centimeters and 1,360 to 1,420 reciprocal centimeters. These results demonstrate that the structural differences between benzoic acid and sodium benzoate can be clearly identified using FTIR spectroscopy. Next, we compared the FTIR spectra of sodium benzoate and the precipitated product obtained after acid treatment. The acid precipitate was prepared through the following procedure. In the sodium benzoate spectrum, characteristic peaks of the carboxylate structure were observed. However, in the spectrum of the precipitated product, those peaks disappeared, while a broad OH bond peak and a CO double bond peak newly appeared. These observations confirm that sodium benzoate was protonated and converted into the benzoic acid form. Through UV visible and FTIR analysis, we confirmed the conversion between benzoic acid and benzoate ion depending on pH changes. In UV visible, changes in the absorption spectra were observed, while in FTIR, structural changes were confirmed through the CO double bond, OH bond, and COO peaks. These results indicate that benzoic acid was converted into benzoate ion under basic conditions.